So I guess, is everyone back? Are we missing anybody? You guys ready? Okay. All right. So now we're going to start the second lecture. We're going to go over the hypothalamus and the pituitary gland. Again, come on thing. Here are the resource materials I use to prepare the lecture, either electronic or in book form. Here are the objectives. Again, please read through them as you study the lecture for your exams and assessments. All right. All right. So this cartoon here is to remind you that the hypothalamus and the pituitary gland are like a master gland. So the hypothalamus is integrating input from higher brain's functions, particularly the limbic system, which regulates our behavioral, eating behavior, reproductive behaviors, and our emotions. It's also going to integrate information from autonomic nerve centers. So it's going to look and see what is the nutrient levels in our blood, how much glucose I have, how much amino acids, what's my body temperature, what's the osmolarity in my blood, what's my the blood pressure. It's going to integrate all those responses, and then it's going to send input into the pituitary gland to have it synthesize hormones which are going to regulate homeostasis. Right? So in the pituitary gland, we have many different hormones. We have both the posterior pituitary and anterior pituitary. Here are the proteins and the functions it's going to work on. So the posterior pituitary is going to secretes vasopressin, which is regulating water balance, and oxytocin, which helps in milk secretion. So it stimulates muscle contraction in the mammary glands for milk secretion. In the anterior pituitary, we're going to stimulate the thyroid hormone, which is going to regulate metabolic rate. It's responsible for responses to stress. We're going to stimulate glucocorticoids, which is responsible for our responses to hyperglycemia, trauma, burns. It's going to stimulate hormones which are involved in reproduction, so our reproductive glands. It's going to increase secretion of hormones that are involved in growth hormone, insulin-like growth factor, and lactation, for instance, prolactin. The pituitary, that thing out of the way. Sorry about that. I didn't know that was in the middle of this everything. All right. The pituitary hormones gland also secretes hormones which have tropic effects on different endocrine glands. For instance, thyroid stimulating hormone is going to activate proliferation in the thyroid gland. Adrenocorticotropin hormone is going to stimulate proliferation of the adrenal glands. And then there's follicle stimulating hormone and luteinizing hormone, which is going to stimulate the production or proliferation of the productive glands. I don't know why that thing does not work. All right. So this is a view of the hypothalamus. This is a sagittal view to the brain, right? And this is a blow up in this cartoon. As you can see that the hypothalamus is above the brainstem and below the thalamus, hence hypothalamus. The hypothalamus is part of the diencephalon. This is the forebrain, the posterior part of the forebrain. You can see that the hypothalamus is functionally linked to the pituitary gland through something called the pituitary stalk. And the hypothalamus is going to secrete hormones which regulates the secretions from the pituitary gland. So basically, the hypothalamus acts as a bridge between the central nervous system and the endocrine system. In this particular image, I'm showing you a coronal cut through the brain. Right? So here you see the hippocampus, the hypothalamus. This brown material around here is the different nuclei or groups of neurons which make up the hypothalamus. This little thing here, that's the third ventricle. And here's the media eminence, which is the lower portion of the hypothalamus. Now, I think we've all know that in the brain capillaries have a lot of tight junctions, and they're very impermeable to transport of water soluble molecules into the brain, which forms the blood-brain barrier. However, around the hypothalamus and the ventricles and the media eminence, those capillaries are festrated. They have holes in them, right? Here in this cartoon, I'm showing you what a festrated capillary is. You see here's the endothelial cells, and there's little holes in them. And this blow up of this electron micrograph, you can see another festrated capillary, and the arrows point to the holes in the capillaries. So what does that allow? So this allows the hypothalamus to communicate with the peripheral circulation. So it allows the uptake of hypothalamic hormones into the hyposphere portal system. It also allows the detection of blood-borne molecules. It can now the hypothalamus can tell you how much glucose is in the blood system, how much amino acids, what's the fat levels. It can also respond to different hormones like insulin and leptin. In this cartoon, I'm showing you the 3 different ways the hypothalamus controls endocrine signaling. At first, step 1, it's going to release either releasing hormones or inhibitory hormones which act on another cell type in anterior pituitary to release a second hormone that regulates cell function. Second, there are neurons in the hypothalamus which extend their exons down into the posterior pituitary. So this is neuronal in that, and they're going to release different hormones directly in the peripheral circulation. These hormones are oxytocin, and ADH stands for antidiuretic hormone, which is also called vasopressin. And finally, the hypothalamus has these neurons which extend all the way down into the adrenal gland in the adrenal medulla, and it directly innervates these chromaffin cells. These chromaffin cells release, in response to that input, release epinephrine and norepinephrine. This is very important when I want to raise glucose really quickly in my blood system. In this cartoon, I'm showing you the hypothalamus, and it's being connected to the pituitary gland. Here's the immediate imminence. You see it's connected to the pituitary and through this pituitary stalk. That stalk is made up of an inner core called the infundibular stalk. This is neuronal material. It comes from the hypothalamus, goes into the posterior pituitary. And around that internal core is the pars tubulus, and this forms the connection to anterior pituitary. We see that the pituitary gland is situated in this bone, is a cavity. It's called a stella tunica. This translates into this Turkish saddle for some reason. I don't know why, but that's what it's called. And it's in the cephoid bone. This helps protect that pituitary gland. All right. So over here we have the anterior pituitary. In some books, it's also called the adrenal hypophysis. This contains the endocrine glands, endocrine cells that respond to the releasing factors from the hypothalamus. And the releasing factors are going to be secreted into the medians, and they're going to be transported to the anterior pituitary to something called the hyposphere portal circulation. And we'll talk about that in a little bit. On the other side is the posterior pituitary. This is also called the neuronal hypophysis. Right? This is neuronal in origin. It's made up of the axons from the hypothalamic neurons that extend their axons down into the posterior pituitary. And these nerve terminals release hormones directly into the blood. And here we have a blow up. This is a histological section through that pituitary gland. I'm not going to ask you any histology questions, but I think just notice that there's a difference between the posterior pituitary to anterior pituitary. The one thing I'm going to highlight is the intermediate pituitary. This is usually atrophied in humans, adult humans, but it's going to play an important part in Addison's disease because it secretes a hormone called melanocyte-stimulating hormone. This is going to give us the brown pigments when you have Addison's disease, which we'll talk about with the adrenal gland next week. So now we're going to follow either the posterior pituitary and anterior pituitary and how these signaling pathways go on. So in the hypothalamus, there are neurons in either the paraventricular nucleus or the suboptic nucleus. These large-bodied are magnocellular neurons. These neurons are going to synthesize and secrete 2 hormones, like we talked about earlier, antidiuretic hormone, ADH, which is also called arginine vasopressin, and oxytocin. They extend their axons all the way down through the glandular stem into the posterior pituitary. Those hormones are going to be secretory vesicle containing hormones are going to be trafficked down the axons, and they're going to be served the nerve terminals in the posterior pituitary. And upon the right stimuli, they're going to be released in the systemic circulation. All right. So here are the structures of both of those hormones. I don't expect you to memorize the structure. They're both 9 amino acid peptide hormones. You see that arginine vasopressin is called arginine because this is an arginine right here. In other species, this is a lysine. For instance, in porcines, it's lysine arginine, lysine instead of an arginine. And below, I'm showing you the target tissues. So oxytocin is going to cause contraction of the smooth muscle in the uterus. This is important during childbirth. It's also going to cause contraction of the in the mammary gland, which is important for producing milk during breastfeeding. Whereas vasopressin has many targets, but the one we're going to be more interested in is its effects on the kidney. And here it's going to act on the collecting ducts of the kidney to increase water reabsorption and make more concentrated urine. In this particular, I'm showing you a schematic of the kidney. And arginine is going to affect water permeability from the distal convoluted tubule all the way down to the collecting duct. Right? So it's going to decrease water excretion. I'm going to increase water absorption, and I'm going to make osmotically concentrated urine. So here's how it works. So in this particular picture, I'm showing a principal cell of the collecting duct. In the sinusoidal side, I have a vasopressin receptor. It's a V2 receptor. When arginine vasopressin binds to the receptor, I'm going to activate the G-alpha S, the stimulatory subunit. It's going to activate adenylate cyclase. I'm going to produce cyclic AMP, and cyclic AMP is going to activate this kinase called protein kinase A. Protein kinase A is going to phosphate many different targets. One of the targets that's going to phosphate are these water channels called aquaporin. This is aquaporin 2. When it phosphorylates that, it's going to increase its excitosis into the apical membrane. This is the lumen side, which is where the urine is. Those water channels are going to increase in here, and I'm going to allow water to come back into the principal cell. It's going to flow across and go back into the interstitium into the blood system. These aquaporin 3 and 4, different isoforms, these are constitutively active. So that's going to be the short term. In long term, protein kinase A can also phosphate a transcription factor called CREB. That stands for cyclic AMP response element binding protein. This is a transcription factor. When it's phosphated, it can go into the nucleus. Actually, I think it's already in the nucleus. So pKa actually goes in the nucleus, phosphates that transcription factor. It's going to bind to different regulatory elements and turn on gene transcription. One of the things it's going to turn on is the synthesis of the water channel, the aquaporin 2. So at the end of the day, I have more aquaporin in the apical membrane. I'm going to bring water into the blood system. And that's how I'm going to concentrate the urine over here and bring up the water volume over here. All right. So what regulates vasopressin secretion? So the main regulator is changes in osmolarity. So there are certain neurons in the hypothalamus that have osmoreceptors, right? And they respond to changes in the osmolarity in the extracellular fluid. If I get an increase in osmolarity, they're going to send messages to either the paraventricular neurons or the suboptic neurons, and they're going to stimulate those to secrete vasopressin. There's also a secondary change. So there's high hemodynamic control. So in both the carotid sinus and the aortic arch, there are baroreceptors, and those are, when they fire, they're putting an inhibitory effect on the paraventricular neurons and the suboptic neurons. So when I have a decrease in blood pressure or blood volume, those baroreceptors stop firing. And so I'm going to de-inhibit that inhibitory signal. Right? So that's the second. But osmolarity is the most important in determining vasopressin secretion. All right. So in these graphs here, I'm trying to illustrate the amount of vasopressin in the blood versus blood osmolarity. And you can see that it's fairly constant up to a point. There's a threshold, about 280 milliosmoles. And when we get above that threshold, there's a linear increase in the amount of vasopressin in the blood. This is due to increase of secretion. In the bottom, I'm trying to show you the relationship from vasopressin in the plasma versus blood volume depletion. And you notice that large changes, like a 10 to 15% decrease in blood volume, is going to stimulate secretion of vasopressin. And notice the difference in the y-axis. This is 0 to 12. This is 0 to 50. So you get a much bigger vasopressin secretion when you have blood volume. And this can concur with either hemorrhagic, which is a decrease in blood, or hypovolemic shock, which is a decrease in fluid. The other thing about vasopressin secretion is you can have additive effects, integration. So in the black line, I'm showing the normal response to changes in osmolarity. So above 200 milliosmoles, you get a stimulation of vasopressin secretion. If I have a loss in blood pressure or a loss in blood volume, you see that it's going to shift it to the left. It's going to become more sensitive. I'm going to shift the threshold to a different level, to a lower level. And this line is much more steeper. So it allows me the system to respond more rapidly. Did that come from here or that? All right. Second thing on this graph, notice this little purple box here. This is when your thirst centers, when you feel thirsty. You notice that you're thirsty only after your vasopressin starting to increase. So only after osmolarity already increasing, when you actually feel thirst. That's why when you're out in the desert, they always tell you to drink even when you don't feel thirsty, because it's kind of late in the response. All right, so here are the feedback loops that control blood osmolarity. So if I get an increase
Professor, I have a quick question. So the last point you said, so your thirst receptors are stimulated after the plasma osmolarity has increased already?
So here, your plasma osmolarity is already increasing, right? So this is somewhere in the neighborhood of about 290 milliosmoles. So I've already increased quite a bit. Vasopressin secretion has already kicked in to try to bring down osmolarity before you even start to think that you're thirsty. So it's not as so the vasopressin system is already turned on before you actually feel the need to drink. That makes sense?
Got it. So when you lose the water, is it coming from the intracellular into the extracellular environment to when the osmolarity is increasing?
How's osmolarity? So how is it? So usually it's probably too much water excretion through the kidney would be one of the ways.
Okay.
You could also have too much salt intake.
Got it.
There's several different ways they can work. All right.
Got it.
All right, thank you. All right. So here are the feedback loops controlling blood osmolarity. So as we talked about, we get an increase in blood osmolarity. It's going to stimulate the osmoreceptors of the hypothalamus. They're going to increase vasopressin secretion. I'm going to increase the permeability of the collecting ducts. I'm going to reabsorb water from the urine, and that's going to result in a decrease in blood osmolarity. And that decrease in blood osmolarity is a negative feedback to turn off the osmoreceptors. The osmoreceptors can also feed forward to the thirst centers in the hypothalamus. That and a drop in extracellular fluid volume can all stimulate the thirst centers. The thirst centers can say, I'm thirsty, I'm going to drink, I'm going to increase more water. By increasing my water intake, I'm going to both decrease the blood osmolality and I'm going to feed back and inhibit the thirst centers. So these feedback loops help to maintain blood osmolarity in the correct physiological parameters. All right. Here's a chart, basically going over everything I just said for you guys to help and you're studying. So where the location of the different receptors, what they respond to. I don't think I remember mentioned this. So the osmolar receptors actually respond to cell shrinkage. The receptor notices how big the surface area is. Here again, what is required for the activation. And here's how much vasopressin is going to be secreted. So with the plasma osmolarity, it's a very small change in vasopressin where you have if you have a huge loss of blood or blood volume, you're going to have a large secretion of vasopressin. All right. All right. So there are some diseases involved with vasopressin system. This is called diabetes insipidus. And this is characterized by the secretion of a large volume of dilute urine and excessive thirst. So there are several causes. One can be a decreased vasopressin release. This is usually caused by diseases in that hypothalamic posterior pituitary axis. Sometimes it's caused by trauma, inflammation, or infection. It can also be a decreased responsive of the kidney to vasopressin. There are known mutations in the V2 receptor, which cause it to be less active. It can also be induced. So low plasma potassium. So low plasma potassium actually stimulates the degradation of the aqua tube channel. So we don't get as much water taken up from the lumen and diluting the concentrating the urine. And finally, you can have excessive water intake. So if you're drinking a lot of water, you can actually get something that's close to diabetes insipidus, but this is not caused by any dysfunction in the vasopressin regulatory system. Right. Now we're going to switch to the anterior pituitary. I know this is a very busy slide. I'm going to go through the things that are really important. First of all, we have this superior hypophyseal artery flowing into the pituitary stalk. It's going to branch out and make capillaries. Those capillaries are going to recoalesce and form the hypophyseal portal circulation. And that portal circulation is going to flow through the anterior pituitary. So the anterior pituitary is being oxygenated by venous blood. There's only one other organ that's oxygenated by venous blood. That's the liver. So very, very unusual. All right. So we have axons in the hypothalamus that are going to extend down to the medium intimates. They're going to release hormones into this area. It's going to be picked up by those capillaries because the capillaries are festrated and it allows the uptake of those hypothalamic hormones. The hypothalamic hormones are going to go into the hypophyseal portal circulation and they're going to flow down and they're going to affect different endocrine cells in the anterior pituitary. All right. Now, another thing to notice that this system is closed, and so these endocrine cells in the anterior pituitary are seeing much higher concentrations of hormones than they would in this normal circulation, because this does not go into the general circulation before it hits their, right? So not diluted. Those hypothalamic releasing hormones are going to stimulate the secretion of tropic hormones, and those I list here. We have, we're going to go through each one of these a little bit later. We have thyroid stimulating hormone, adrenocorticotropin hormone, follicle stimulating hormone, luteinizing hormone, prolactin, and growth hormone. And below, we're going to show all the target tissues. We'll go through that in a little bit later. The other thing that's important is notice that they see that this is feedback here. So those hormones can feedback at the level of the anterior pituitary or the hypothalamus to turn off and control secretion. In this particular chart, I'm listing all the hypothalamic hormones, what target cell they act on in the anterior pituitary, what they do in the anterior pituitary, and what the principal action of that anterior pituitary hormone is. So we're just going to go through and I'll list them. I know it's kind of boring, but make sure everybody on the same page. So we have, and then we notice that they're all more or less named after the cell types they're activating on. So we have thyrotropin releasing hormone. It's going to act on thyrotropes. It's going to stimulate that cell to increase the secretion of thyroid stimulating hormone. And thyroid stimulating hormone is going to be secreted in the general circulation. It's going to go to the thyroid gland when it binds to the receptor. It's going to stimulate the secretion of thyroid hormones and the growth of the thyroid gland. Thyrotropin-releasing hormone also acts on lactotrophs. It's going to increase the secretion of prolactin. This is important for the production of milk. Gonadotropin-releasing hormone acts on gonadotropes. It's going to stimulate the secretion of luteinizing hormone or follicle-stimulating hormone. Luteinizing hormone is involved in stimulation of ovulation and luteinization in females, or testosterone secretion in males. Follicle-stimulating hormone is going to activate ovarian follicle growth and estrogen secretion in females, and whereas it's going to stimulate somatogenesis in males. Then we have corticotropin-releasing hormone. It's going to act on corticotrophs. It's going to stimulate the release of adrenocorticotropin hormone, or ACTH. This hormone goes to the adrenal gland, where it's going to stimulate the production of glucocorticoids and the growth of the adrenal cortex. Then we have growth hormone-releasing hormone. It's going to act on somatotrophs. It's going to stimulate the secretion of growth hormone. Growth hormone goes to the periphery. It's going to stimulate the production of IGF-1 and an increase in body growth. And then we have the inhibitory hormones. We have somatostatin, which acts on somatotrophs and thyrotrophs. It's going to decrease growth hormone secretion and also decrease thyroid stimulating hormone secretion. Then we have dopamine, which acts on lactotrophs and thyrotrophs. It's going to inhibit the secretion of prolactin and inhibit the secretion of thyroid stimulating hormone. All right, I always say a big list, but it's something you got to memorize. You need to know this. Now, one of the most common hormone secreting tumors is one that secretes prolactin. Now, just looking at this chart, what would you guys think would be one of the treatments for a person who has a tumor that secretes prolactin? These are called prolactomas. So what would you, if you were the doctor, what would you be prescribing? Anybody online or in the audience?
Give them dopamine.
Yeah. A synthetic dopamine, a long-lasting dopamine would be one of the first-line treatments for someone who has a tumor like that. And after that would be surgery. So these things are very important if you think about it. If you know what these hypothalamic hormones are doing, you can adjust and you can make therapeutics out of them. All right. Now I'm going to go a little bit into growth hormone. As I said before, growth hormone is a peptide hormone. It's going to be produced by the somatotrophs to anterior pituitary. It is processed and synthesized like all other peptide hormones through the secretory pathway. The one thing interesting about growth hormone is not really dissolvable in water. So a lot of it's going to be bound to growth hormone binding protein. And the interesting about that is that the growth hormone binding protein is actually generated by a proteolytic cleavage of the growth hormone receptor itself. So it's cleaved, that's the binding protein. So at the level of target tissue, this growth hormone binding protein can actually compete with the target tissue for growth hormone because it's got the same affinity as a hormone receptor. In this cartoon, I'm showing you the regulation of growth hormone secretion. The primary regulators of growth hormone secretion are growth hormone releasing hormone, which is going to act on the somatotrophs to stimulate growth hormone release. And the inhibitory factor is somatostatin, which is going to inhibit growth hormone release. We also have other inputs that are important. Hyperglycemia is going to inhibit growth hormone release, whereas amino acids are going to stimulate growth hormone release. These amino acids are particular branch chains like arginine and leucine. We also see we have a short circuit with growth, like for instance, growth hormone releasing hormone can actually feed back to the hypothalamus and inhibit its own secretion. And we have a long loop where growth hormone itself can feed back at the hypothalamus and inhibit the release of growth hormone and releasing hormone. Now, growth hormone has some direct effects on peripheral tissue. Did you have a question? Oh, sorry. So growth hormone can have direct effects on peripheral tissues, but one of the biggest effects it has is at the level of the liver, where it's going to stimulate the liver to generate another hormone called somatostatin. This is also insulin-like growth factor. Somatostatin is the mediator of somatic effects of growth hormone. That's what it stands for. Some of the old books have that. And below, I'm going to show you all the targets of growth hormone and insulin-like growth factor, but I'm going to talk about those in a little bit later. And you notice that IGF also has an inhibitory factor. It can feed back at the level of the hypothalamus to decrease growth hormone releasing hormone secretion. It also enhances somatostatin secretion. It can also feed back and directly inhibit growth hormone release at the level of the pituitary, at the level of the somatotropes. So here's the signal transduction pathway. So growth hormone releasing hormone binds to its receptor and it's going to activate adenylate cyclase, going to increase cyclase AMP, activate protein kinase A, which is going to phosphorylate a calcium channel. That phosphorylated calcium is going to allow more calcium into the cell, and that's the trigger for vesicle secretion of growth hormone. The somatostatin receptor is coupled to G-alpha I, is going to inhibit adenylate cyclase, and that's how you shut off this pathway. You're going to decrease PKA activity, you're going to no longer phosphorylate the channel, and you're going to decrease the growth hormones release. All right. The other thing that's interesting, important about growth hormone is that it's cyclic. So it isn't trained to sleep. You know, in this particular graph, the highest amount of growth hormone is at night when you have the onset of deep sleep. The other thing that's important is that it's more active, more secretion during pubertal growth versus when you get older. So the level of hormone is smaller in the adults versus someone who's 14 years old. Right. Here again is a chart telling you exactly what I just talked about. What are the main inhibitors and stimulators of growth hormone release? So we talk growth hormone releasing hormone, somatostatin. The inhibitors also includes there's some input from insulin-like growth factor, hyperglycemia. Talk again about the amino acids stimulating growth hormone and deep sleep. Right. So how does growth hormone signal and produce a biological response? So growth hormone is going to bind to its receptor. This is a receptor link kinase. There's these different kinases involved. They're called JAK or Janus kinase. Again, binding the hormone receptor activates the kinase, which is going to phosphorylate this transcription factor called STAT. STAT stands for signal transducer and activator of transcription. Once it's phosphorylated, it can dimerize. That dimerized transcription factor can now translocate into the nucleus, where it binds to regulatory elements on genes and turn on gene transcription. One of the things it's going to turn on is going to be the production of insulin-like growth factor 1 and the binding protein for insulin-like growth factor 1. And those two factors are going to regulate growth and development. Growth hormone can also induce its own turnoff. So it's going to stimulate the expression of this protein called stands for so it's inhibitor. It's an inhibitor of the JAK kinase. So this particular protein is made. It's a negative feedback effect on the so it shuts down this pathway. So it can both generate a IGF-1 and also turn off the pathway in the same direction. All right. So what are the effects of growth hormone? So growth hormone can have direct effects on tissues. It has a direct effect on adipose tissue. It's going to stimulate lipolysis and decrease whole body adiposity. It also has direct effects on the liver. Like I said earlier, it's going to stimulate the production of IGF-1 and the IGF-1 binding protein. Now, this IGF-1 probably mediates most of the somatic effects of growth hormone. So if you get rid of this, you don't have a lot of the effects of growth hormone. So most likely it's IGF-1. I forgot also insulin growth also stimulates hepatic glucose. It increases gluconeogenesis in the liver. So IGF-1, maybe also growth hormone, has effects on linear growth. It acts on both bone cells and chondrocytes or cartilage cells. It's going to increase protein uptake and increase protein synthesis, so it's proliferative. It also has effects on muscle. It's got anabolic effects. It's going to increase protein uptake and increase protein synthesis, and again, decrease glucose uptake. And finally, it's going to have effects on many different visceral organs. It's going to increase the proliferation of your heart, your kidney, your stomach, and your intestines, and increase organ size. The other thing to know about growth hormones, it's got an anti-insulin effect. So in muscle, you notice that it decreases glucose uptake, whereas insulin will stimulate glucose uptake. In adipose tissue, it's going to decrease glucose uptake and increase lipolysis, whereas in the insulin, it's going to decrease lipolysis. In the liver, it's going to stimulate gluconeogenesis, whereas insulin is going to inhibit gluconeogenesis and stimulate the storage of glucose. And in both liver, fat, and muscle, it's going to, when a growth hormone binds to those receptors in those tissues, it's going to make them more insulin resistant, so they can't respond to insulin. So the net effect that growth hormones is going to shift the body's metabolism from glucose utilization to lipid oxidation. Right. So in this particular cartoon, I'm showing you the different hormones which are responsible for the growth of an individual. And the size of it is relative to their importance. You see at the early ages before 2, both growth hormone and thyroid hormones are responsible for the growth. Thyroid hormones actually stimulate the expression of growth hormone. See, growth hormone is important until you reach about toddler age, and then it starts to decline. And when you reach puberty, both growth hormones, thyroid hormones, growth hormones, and the androgen hormones are responsible for that large growth during puberty. Right. So what happens when growth hormone secretion goes wrong? So if I have a deficit in growth hormone secretion, I generate something called pituitary dwarfism. These people they produce a small person, but the proportions are normal. This is a person with a pituitary dwarfism. This person here is someone with a giantism. The same to give you an idea of the difference in the size. So if you have growth hormone secretion that goes excess when you're during childhood, we develop a disease called giantism. In this particular picture, here's a normal individual. Here's his sibling, which has excess of growth hormone secretion. You notice that one of the things that, since the long bones are the epithelial plates, the long bones are not sealed, you have a much longer arms, longer feet, proliferation of increase of the long bones. You have bigger hands and bigger feet. If I have growth hormone secretion as an adult, here are the long bones, the epithelial plaques and the long bones are already sealed. This is after puberty, so it's not going to involve long bones. So you can see in this picture, this is the person with acromegalia, which is hypersecretion of growth hormone in adults. You see as she ages, you see the coarseness of her jaw getting thicker, her tongue getting thicker, her coarse hair. Here's the characteristics. We get a thickening of the bones of the hands and jaws and the feet. We get a enlargement of the jaw and the brow, increased brow, increase in the tongue. The hair becomes more coarser, and you get an overdevelopment of the muscles. We can also have cardiovascular problems and renal problems, as well as diabetes, because there's no, we have stimulation of hepatic glucose production by the liver in response to growth hormone. Right. Right. So the mainstay for this treatment is usually surgery, and somatostatin analog was usually going to inhibit growth hormone secretion. Right. Right. That ends that particular lecture. Are there any questions? Either in the house or online.
Professor, I have a quick question. We discussed how growth hormone stimulates organ size. Is this talking about like increased cell division, or just the cell size itself getting bigger?
I think it's going to, I don't know if it's a good question. I don't know for sure. I think it'd be both of those.
Okay. Because I was wondering, could this be like stimulated to, I guess, improve the health of like a damaged or organ or something?
Say again? I didn't quite catch that.
I was thinking that could this be stimulated to fix a damaged organ or something that's not functional right now?
That I do not know.
Got it.
Good question. I have no idea.
Right.
I mean, you do give growth hormone to people who are small in size to help them. So if you're not reaching your target height, people do get growth hormone treatment to try to increase their longitudinal growth. So it can stimulate that. Whether or not it changes the size of the visceral organs, if they're damaged, I don't recall.
Okay.
Right.
Got it. Thank you.
You're welcome. All right. Again, I have some typical questions that might be on exams. We'll go through this way quick. All right. So circulating level of which of the following pituitary hormones would be unaffected by interrupting the blood flow through the medium elements? So here are these different hormones. Which one would not care if we block the blood flow in the medium elements? Any idea?